Hi everyone, welcome to the Biodiscovery from Composting Systems Living Laboratory. We're located at the Environmental Engineering Field Site on the Northern Arizona University campus in Flagstaff, Arizona. My name is Jeff Meilander, and I'm a research scientist in the Capareso Lab, where I lead the Compost Research and Applications program. We're currently running 2 experiments right now. The first focuses on human excrement composting, and the second is a vermicomposting system that we've recently launched thanks to funding from the NAU Green Fund. We're an eclectic mix of researchers and graduate and undergraduate students who believe that composting is cool, useful, important, and transformative. Let me show you around. But first, let's take a minute and meet some of our compost crew.
I'm Greg Capareso, professor of biology at Northern Arizona University and also co-director of the Pathogen and Microbiome Institute at Northern Arizona University.
Hello, my name is Olivia Garcia. My major is bioinformatics and I'm the I2S student. Interns to Scholars, it's a program we have on campus where we can intern and just get more experience in our field and work with a mentor.
Hello. I'm Liz Gehret, and I'm a senior research software engineer in the Capareso Lab.
Hello, my name is Kaylee Kashner. I'm a sophomore, and I'm majoring in environmental science with an emphasis in biology.
My name is Sam Lockhart. I'm a PhD student in astronomy and planetary science, and I'm basically here studying microbes and how these can potentially relate to microbes in the cosmos.
Here you can see 8 composting bins constructed from recycled pallets. Each bin is lined with recycled billboard vinyl to prevent any contamination from leaching out into the soil below. The source material that we used for our human excrement composting system came from a truly local experience.
So Jeff comes to me and the other people in the lab and is trying to figure out how we can acquire mass quantities of fecal material. In any individual trip, people poop into large ammo cans called groovers that have a toilet seat on top of them. And at the end of the trip, all of these filled groover cans are then provided to Chris Sonifrak, and he owns Groover Kinda Love. This is the guy who disposes of all of the poop that's accumulated on each Grand Canyon river trip. So this summer, Chris came by and delivered 250 Groover cans to get us started on this project.
As you can see behind me, Flagstaff is surrounded by the largest contiguous ponderosa pine forest in the world. So using ponderosa pine chips was a great option as our bulking material. Before adding the excrement, we collected a mix of pine chips, shavings, and needles, and added about 15 centimeters of this material to the base of each bin to absorb moisture and leachate during the composting process. To optimize conditions for microbial activity, the bulking material was lightly sprayed with water to reach a moisture content of 45 to 65%, which is ideal for thermophilic composting.
We see human excrement management as a growing global challenge, but also as an opportunity. Extreme weather events like flooding and rising sea levels are threatening the functionality of wastewater treatment plants and septic systems. Additionally, as the world's population increases, billions of people still lack access to safe sanitation. And when combined with open defecation, this leads to environmental contamination, pathogen exposure, and ultimately human mortality.
Composting human excrement isn't going to save the world, but it can be an integral part of treating human waste. And we think that we can do this through composting by diverting excrement before it gets sent to wastewater treatment plants. After the initial wood chips were placed in the bottom of the bins, 4 to 6 groovers of excrement were added, and we collected 3 samples from random locations within the excrement layer using a screw cap spoon attached to a 30 milliliter fecal container. Next, we added wood chips and homogenized the material within the bin to evenly blend the material and support key composting parameters, doing our best to target a carbon to nitrogen ratio between 20 and 40 to 1, a moisture content of 45 to 65%, and provide sufficient aeration. This process was repeated 4 times, resulting in the addition of roughly 16 to 24 groovers to each composting bin and a pooled conical tube, creating a composite fecal sample representative of that bin. Once the bin was filled, we leveled the surface and added about 15 centimeters of wood chips to serve as a biofilter to help reduce odors, retain moisture, and insulate the pile. Thanks a lot, Chris. We really appreciate your help. From there, we recorded near daily temperatures and collected weekly samples for a range of microbiological analyses. So we are going to be sampling from each of our piles. At the very beginning, we take our core temperature to get down into the center of our pile. And then we dig a hole about the size of our trowel in the shape of a 5 on a die. And this pattern is actually known as a quincunx. Then we take temperatures from each of those holes with a meat thermometer so that we can get an average temperature for where we're sampling. And once we've completed that, then we grab our little sifter and our bag that's marked with our identification, and we're going to take a little sample from each of these holes, and we're going to put that into our sifter, and we're going to shake this and fill up our bag about halfway. So once we've got our bag filled, we're going to homogenize the material again just to make sure we mix this up, and when we sample from here, we're getting a little bit more of a representative sample out of here. After 20 weeks of sampling, we will perform DNA extractions, culture for E. coli, and run qPCR assays targeting E. coli, Clostridium perfringens, adenovirus, norovirus, and a broad range of enteroviruses. To characterize microbial diversity, we'll conduct 16S, ITS, and 18S amplicon sequencing to identify bacterial, fungal, and eukaryotic communities. In addition, metagenomic analyses will be used to profile all the genetic material within the samples, while metatranscriptomic analyses will reveal functional genes involved in enzyme production and other processes contributing to composting activity. We'll also analyze the samples for nutrient content and eventually pharmaceutical content. Once this is complete, our lab's data scientist and bioinformatician, Dr. Chloe Herman, will use QIIME2 to begin to make sense of all this data. She will also be training current and future undergraduate students involved in our project.
Oh, hi. I didn't see you there. My name is Chloe Herman, and I'm a data scientist in the Capareso lab with a PhD in bioinformatics. I'm so excited about our human excrement composting project because of the impacts it can have on soil restoration and agricultural production. I'm also really excited to explore the fungal and microbial communities necessary to turn this human excrement into nutrient-dense compost.
Initially, we only turn the bins once temperatures drop below 55 degrees Celsius. When that happened, we remove the pallet door and spread the compost out across the tarp. We would move the outer material towards the center and the inner material toward the edges. This ensured that as much material as possible was exposed to thermophilic temperatures. Turning the compost showed a steady decomposition, though toilet paper broke down slowly and some contaminants like flushable wipes and feminine products remained intact. Turning also helped us identify clumps of excrement that needed to be broken apart, fungal growth, and areas where moisture levels were uneven, typically drier along the edges and wetter near the bottom. If the piles no longer reheated above 55 degrees Celsius after a major turning event, we switched to using a compost crank to aerate and mix the material. We continued this maintenance process every Tuesday after sampling to keep oxygen levels high and support microbial activity. And here's some of our finished product, which will rest over the winter before being used in new experiments this spring. Now, as I mentioned earlier, we recently launched a vermicomposting program that uses food waste collected from campus dining halls.
Look at these worms! Awesome! Wow! Good luck, worms!
Hello everyone, my name is Charlotte Walker Hurley and I am a biology major at Northern Arizona University. I currently am the undergraduate research manager for the vermicomposting project, where I help oversee the experiment and weekly data collection focused on sustainable waste management through the use of red wigglers.
We're excited to be hosting a celebration on World Toilet Day right here at our site to showcase our research and raise awareness across NAU's campus and the Flagstaff community about global sanitation issues. The event will feature a walking tour and informational pamphlet highlighting our work, global sanitation issues, and sustainable solutions like composting toilets, urine diversion, and wastewater treatment. Looking into the future, our site is becoming a hub for innovation in composting, microbiome science, and sustainable sanitation.
First, we're exploring space and beyond, testing how plants grow in Martian and lunar regolith using human excrement compost, helping us think about off-world agriculture.
Next, we plan to do spike-it experiments testing the degradation of pathogens and pharmaceuticals to better understand compost safety and contaminant removal.
We're also developing off-grid research capabilities with a site designed for sampling, DNA extractions, sequencing with MinION, and on-site analysis. In other words, we're working toward bringing the lab wherever it's needed.
Using omics technologies, we hope to build synthetic microbial communities that can act as inoculants that can improve composting efficiency and performance.
Beyond sustainability, I am also interested in the research potential of vermicomposting, particularly in its ability to help us understand how pathogens interact and break down within composting systems. This kind of research could one day play a key role in making human waste composting safer and more efficient.
We'd love for you to get involved through collaborative research, student engagement, or shared mentorship, and help us advance microbiome and sustainability research together.
Have a great conference. Thanks for joining.